Hello everybody. So, welcome back where we finished last time. So, in this class we will talk now after 1, 2 we will go for the type 3. So, type 3 copper proteins where we can see these. So, the nature of the center already we have learnt it, we have seen on some other examples, but again we can just concisely again we can see some new examples, some more things, all these things. So, there are coupled dinuclear, not uncoupled long distance copper centers. So, they are coupled, they are nearby and they are responsible for O2 activation again, but you require 2 copper centers, unlike your problem that we have seen earlier in case of your galactose oxidase. You have to oxidize the protein part or the ligand part to get the electron. Then these two important molecules, the tyrosinase activity and the catechol oxidase activity we will see. So, what is tyrosinase? Okay. So, tyrosinase you should know in that way that we know the tyrosine amino acid and ase we all know. So, something we are going with that of your tyrosine residues. So, we will be going for the corresponding hydroxylation or oxidation of this particular enzyme we see on the substrate side tyrosine. So, it is not very huge molecule, is only 42 kilodalton, having again only 2 type 3 centers. So, if you have 2 copper centers like copper 1 and copper 2 and you have the corresponding donor groups L1, L2, L3 and L4 and again one is also 1. So, how you can just bridge these two, because we have seen earlier that hemocyanin is also a good example of your type 3 copper center, but you have only 3 donor groups around copper 1 and another 3 around copper 2. So, what is happening now here? We will be looking for some reactions which are giving you the corresponding hydroxylation as your orthohydroxylation, and that orthohydroxylation of phenols and subsequent oxidation to orthoquinone is very important reaction. That is why we know that in potato skin or any other fruit pulp there is catechol oxidases. Tyrosinase is also responsible for our skin melanin formation. So, what we get? We get some phenol part, you oxidize it to make it a diphenol or bisphenol or catechol. So, phenol can be converted to catechol and catechol can further be converted to your quinone, because quinone is important. For all these cases you have the huge conjugation, that is why it is the color is colored as well as it will absorb the radiation, it will absorb nicely the H nu, the sunlight also. So, like 1, like 2, your 3 we have reached here now 3. So, how schematically you will be able to identify, you should be able to know it. If this site is given to you, identify the nature of the type, you should be able to tell it is not type 1, it is not type 2, it is type 3. So, only the positioning, the detailed positioning of the histidine residues or histidine amino acid residues with that of your imidazole side chain coordinating to your copper center, giving rise to this, and you have the one group what we have seen in this particular box, that is OR, and here it is writing as X. That means you can have many groups over there, you can have the water molecule, you can have the hydroxide group, you can have the alkoxide functions or many other things. So, oxygen uptake can take place 2 copper centers, you are happy with supplying 2 electrons from shuttling of the copper oxidation states between us and IC. So, the distance of these two are important because you are trying to activate that O2, whether this again in the same fashion like that of our hemocyanin, and most of the time people developed all these literature, people studied it, people researched it on this, is that they compared it with the well known hemocyanin molecule. So, with hemocyanin you have, which is a oxygen transport molecule, you see this tyrosinase is a copper enzyme now, but your active site is very much similar. So, electronic transition, the corresponding resonance Raman spectroscopic signature as well as the EPR can tell you or identify the nature of the site what is there in type 3 center. So, the assignment of that particular O2 oxidation state, whether you are with the oxidation state of 1 minus or 2 minus in superoxide or peroxide. So, use the corresponding vibrational stretching frequency measuring technique, which is your FTIR, and here since your O2 is symmetric we will be using resonance Raman technique or RR technique. and also the electronic transitions for to identify the LMCT transition. So, charge transfer transition you can identify, and they extensively you can study it for the last 20, 30 years. We have studied it. That is why the area is matured now, is rich in all these informations, and you can have the more number of informations and which have justified nicely for your transfer also. So, you see that the active site is nothing but your Cu2O2 diamond thing. You have one copper here, another copper here, and you have the two these two fingertips, this is the oxygen. So, the diamond core Cu2O2 or CuO diamond core is important, is a coupled binuclear copper system with a type 3 center. So, these are very important and has been studied biochemically, biophysical measurements, as well as all these informations are supplemented further throughout the understanding about the coordination chemistry, the redox chemistry or non-redox coordination chemistry also. What we see that there are large number of such systems, we all know, and the structural and spectroscopic characterizations basically could be able to establish many such sites, not that with a hemocyanin, but we can identify those sites in the tyrosinase molecule or in the catechol oxidases also. So, what basically they are doing? So, not only the naturally occurring system, but also with that of our synthetically prepared model compounds, what we see now is the model compounds are also very important to tell us many informations. If you take these three examples, Hc is hemocyanin, Ty is the tyrosinase molecule, and CO is not carbon monoxide, but it is catechol oxidase. Why we abbreviate in a particular point in immediately discussing all this? Because you should be able to know that without telling hemocyanin and all big thing, you should know it is Hc. As you know, as Hb is hemoglobin, Mb is myoglobin. So, these three species, they are very big species, but we are abbreviating it is a small term, even less than that of your amino acid residue, where we are using 3 letters, but sometimes the single letter abbreviation is important. So, these three molecules and these particular categories of activation, you can have the mu 1,2 peroxide bridging between these two copper centers, all we know. So, this sort of thing, or you can have these two oxygens coming from these two copper in this particular fashion, the cis binding, two oxygen in my two finger heads and come and bind to two copper centers. Then you can have the corresponding mu eta 2 peroxide bridging, which is well known for your hemocyanin molecule, and the bis oxo. How the oxo is forming, which we are writing as a highly electrophilic in nature, compared to that of your mu 1,2 peroxide 1, which is nucleophilic in nature, because the substitutions, the catalytic functions and all these things are dependent on your nature of this reactivity site, whether it is nucleophilic or electrophilic. So, when you have this mu eta 2 binding between these two copper centers, what you see that at one point, not in your biological world, which is a soft world, but if you go for a very harsh and hard world like your synthetic molecules or laboratory prepared model compound, you can break the O bond. Many model compounds you can have where you can break the O bond, where you have the initially formed this mu eta 2 binding hemocyanin type of molecule. If you break it, the individual oxygen centers can take up more electron from the copper centers, making those two copper centers to the trivalent state, which is very unusual, but sometimes you can get in the synthetic molecules, and you have two oxygen centers which are your oxide centers. Similarly, for the examples what we have seen for your dopamine beta hydroxylase or PHM, we have the mononuclear center and you have the terminal binding like that of your binding in your myoglobin molecule. So, you have the end-on eta 1 binding, which is superoxido, side-on eta 2 binding for superoxido molecule is also possible, end-on peroxido in eta 2 fashion, and end-on eta 1 binding is also possible, which is available for our corresponding superoxido molecule at the left bottom, and then one electron transfer making it peroxido, but immediate proton transfer will make that center as hydroperoxido species, and that can also be functioning as electrophilic center for your all the transformations and the reactions. So, we will talk all three together basically, because they are very much similar. So, side by side it will be very easy to understand the reactivity pattern of all three together. Then what you can do? you just compare which is there in hemocyanin and why hemocyanin is functioning only in this fashion and why tyrosinase is functioning in a different fashion and your catechol oxidase also. So, the name will tell you, CO will tell you that it converts the different types of catechol to its quinone, that is why it is known as catechol oxidase. The electron transfer reaction as well as the removal of your protons, the catechols all having OH function. So, when you is binding to your copper center, your OO are O minus O minus. Then when oxidation is taking place, it is accommodated in such a fashion through semiquinone, it will go to your quinone form. So, this catechol molecule when it is going to the quinone, but in case of tyrosinase we see that is going for the hydroxylation of the monophenol on the tyrosine amino acid is nothing but a phenol part on the amino acid part. So, that basically going for the hydroxylation, the second hydroxylation, already you have one phenol OH, phenolic OH, the second phenolic OH will come through this particular reaction. So, both these TY and CO therefore can go for 2 electron transfer and oxidation of these finally to the quinone. And since you have this 2 electron oxidation, we can also consider it as you are seeing some catecholase activity. So, you should be able to remember it in that fashion that which one is your catecholase activity and where catechol is your substrate. And when the catechol is substrate, 2 electron transfer and 2 proton transfer in the different direction basically giving you the quinone. But in case of tyrosine, it is working on the phenol molecule. So, it will catalyze the conversion of phenols to diphenols. Then phenol is there and the ortho position of that phenol will be hydroxylated. So, the tyrosinase basically when it is converting L-tyrosine molecule, what happens then? So, L-tyrosine molecule you should know about, that is why all these things in terms of your exam, in terms of your questions and all these things you try. How will you remember all these things nicely? Do not go for memorizing blindly. You know, you should know also the L-tyrosine, because all the time I should not give you the chemical structure or the chemical drawing of L-tyrosine, but it should be in your brain. Whenever you reading L-tyrosine, like your alphabet, identification of the alphabet A, B, C, D, we know that how to identify what is A, what is B. Similarly, when you are uttering something, that L-tyrosine, things should be in your mind. From your memory, you should not tell is L-tyrosine. You should be able to know and understand about the structure. Then if I say that something is happening, but the name it tells you that something is related to phenylalanine. So, how it is related? What is alanine then? You should know about alanine, and the phenyl substitution will give you the phenylalanine. That is why you are bringing the phenyl ring on alanine substrate or alanine backbone. That phenyl ring, when it is first time hydroxylated, giving you the paracresol type of molecule. We all know paracresol is you have the methyl function opposite to that of your OH of the phenol ring is 4-methyl phenol or paramethyl phenol is your paracresol. Similarly, you have the substitution, the amino acid is your bigger substitution. So, that bigger amino acid substitution away from that your OH function, that means in the opposite direction is para position, is giving you the L-tyrosine molecule. So, when it is hydroxylated, you are getting adjacent OH groups. So, it is 3,4 dihydroxy L-phenylalanine. So, abbreviated in that way also. D has been taken from the entire name, O is also taken and PA is your phenylalanine. So, you will forming L-dopa is a very useful name. Doctors also know as a drug molecule, and all these things are basically important, and in our brain basically we are producing these important molecules. So, oxidation further of this. So, it is one part of oxidation, that means you are forming tyrosine to L-dopa, then L-dopa will be forming something that to L-dopa Q. Once we know that catechol type of molecule, if it is in your hand, you can immediately oxidize to its quinone form. It is a very simple thing. When you take in the laboratory, when you take it in the laboratory, when the bottle catechol is there, if you open the bottle and keep keep it for some time in air also, the air oxidation can take place, but that amount of dopa Q formation is very less. So, this particular formation is basically catalytic one, and is a huge amount of this will be forming. So, the arrangements what you can have now in your hand, the domains where the subunit structure we should know about, that means for the tyrosinase molecule origin is streptococcal, the streptococcal functions, the streptococcus is important, where you can study all these, you can isolate the tyrosinase molecule from there and the catechol oxidase from sweet potato. So, these are the two sources of these molecules basically, and this similarity in spectral properties basically implies that this hemocyanin should also have a catalytic activity of this type, but it is not showing us. So, the distinction between these two major functions of oxygen transport and oxygen activation in these enzymatic activities determined by the presence and absence of the protein domain covering the active site. So, like your corresponding covalent bond formation in galactose oxidase, you can have something here also happening, such that you can see that it is enzyme, it is not your oxygen binding and transport protein like your hemocyanin. So, hemocyanin behaves as a silent inactive enzyme. So, what you can now consider that if you consider that what is hemocyanin then? Hemocyanin is used for only oxygen binding and oxygen transport, but it is not your enzyme. But it can be activated if you go for the blocking of the amino acid just discarding, that means removal. So, the blocking part, that means the active site access basically is important. If it is not there, it is only functioning as oxygen trapping and oxygen releasing molecule. So, isotopic leveling with oxygen 16 and 17 and even 18 can give rise to all these informations nicely, that the incorporations of the substrate which is taking place over there, the oxygen atom which is getting incorporated within the substrate is coming from this labeled oxygen, which we are using in your O2 molecule as your oxygen gas. So, you have the tyrosine molecule. So, schematically what you see quickly, we will see that if you go for the corresponding thing, that means the corresponding hydroxylation reactions, and that hydroxylation reaction is basically giving rise to the corresponding OH orthoposition to that of the already existing OH molecule. So, what you see now is a bigger description, you can have the simplified description in other way. So, on the right hand top you look at what you know nicely from your hemocyanin days. So, from the right hand top what you see, you have the two copper centers, instead of leveling as copper 1, copper 2, you level it as copper A and copper B is fine, but the overall it is the type 3 center. Then something is happening over there, that is hydrogen bonding, water molecule, all these are nearby, but is not directly coordinating to these two copper centers, but you bring the phenol center. So, that means that particular deoxy form or the lower oxidation form, that means the Q+ form is part of the enzyme. So, when the O2 is getting activated, your phenol can be your substrate like your tyrosine. Tyrosine is also a phenol, substituted phenol, para substituted phenol. To us, for this catalytic activity, phenol is your there and your paraphenol, the para substituted phenol is there, but when it is substituted like tyrosine, it is also a substituted phenol. So, it is basically binding and is try to interact with the copper A center, and you all know that you can have the corresponding binding. So, apical coordination can take place, and that particular copper, you see the copper A is converting to a octahedral geometry. So, not only electron transfer, your change in coordination number is important. So, that is why phenol is coordinating like your galactose oxidase from the top. So, now you have more complex copper center, but your phenol is coming from the top and is coordinating there through this particular site, and only the copper A is involved. So, it is engaged over there. Now, you see that what can happen over there, that this particular dioxygen molecule is there, and that dioxygen molecule is responsible for your formation of the hydroxide of bridging. So, proton is coming from the hydrogen bonded water molecule from here, the lower bottom part. So, this proton basically is coming from this protonation of this particular oxygen of this dioxygen molecule, and then that basically gives us, for the model studies we have also identified that you will be able to break the O bond. So, while you break the O bond, what is happening? In the model compounds we have seen we have the dihydroxide of bleached dicopper system, but once you break it, one is coming out as the hydroxide and it will stay like that bridging part, but the other hydroxide of function, but it is not hydroxide, it is activated oxygen. So, that activated oxygen, if it is peroxide, if we break it, what you are having with? You are having with O and O with a single charge, but again you have the further electron transfer and you go for the corresponding one as the corresponding oxide. If one is oxide and another is radical, and that can also attack the corresponding phenyl ring. So, that activated oxygen, do not bother about other things, that activated oxygen from the top side is basically now attacking your phenyl ring at the ortho position of the phenol. So, it is basically a very good hydroxylating agent. Chemical hydroxylating agents are well known to us. We know metachloroperbenzoic acid, iodosyl benzene and many other are there, like your cytochrome P450, which is ferryle species, iron, oxygen. Why that ferryle species is important? Because that oxygen on iron is a very good reagent for oxotransfer reactions. So, here in situ we are activating all these things, we are not getting any copper double bonded oxygen or copper bound this oxide species, because it is not very well known. People sometimes have that idea also that we can have like ferryle species, we can have the cupryl species. So, once it is got, now your already transformed molecule, that means your catechol part is used as a bridging ligand, a very unique bridging between these two copper centers. One of the phenolate function, one of the phenolate O minus is bridging these two copper center, but another one, the original one is coordinating like that of your apical coordination to that particular copper site. So, you understand in that way, and the elaborate thing is also that the bringing of this water molecule, and water molecule is basically pushed by two other amino acid residues from the bottom and they are hydrogen bonded. So, this water molecule is not a free water molecule. In the biological system, most of the time you see that your water molecule is not free like your test tube water. It is always bound or hydrogen bonded to that particular site. So, when you determine the excess structure, why the protein crystallography is so difficult and so tedious, that you have to identify not only the copper environment, the entire protein part, entire carbon, hydrogen, nitrogen, sulfur and all, as well as the other part which are nearby to that particular active site. So, the deprotonation of a phenolic substrate in the tyrosinase cycle, we are looking here the cycle, that is why is basically devoted for that particular purpose. So, what we see that is the tyrosinases also hydroxylate other monophenols in the ortho position, that is why to diphenols, followed by oxidation to orthoquinones. So, hydroxylation followed by oxidation if we allow, and these two molecules are therefore very similar type, and in one case you see that there is a thioether bond to one of the histidine ligands of CUA, which is by cystinate 92 and histidine 109, like that of your galactose oxidase. So, if you nicely or coolly read the galactose oxidase thing, you now have that particular idea that cystinate anions are so interesting that it can give you new bond to your phenol residue or the phenolate residue. It can also give you the histidine residue. Many other examples are also there. In future also we will take that particular example in different form. So, the hydroxylation of monophenol substrate at the ortho position require a distortion of the Cu2O2, is these two are subscript Cu2O2, that is your diamond thing, Cu2, 2Cu and 2O, and its consequent reorientation of the substrate. What does it mean? That you write like this and you can have the corresponding movement of all these. So, all these movements. So, basically in this particular plane, because copper is fixed. So, this copper basically can allow, so only the movement of the copper is moved there, and this particular, so you can have also this sort of bending also this O. So, these are the things basically. So, these sort of distortions, and at one point it is fixed over there. So, this reorientation sometimes it is allowed and sometimes not. When it is allowed, it will not be possible in catechol oxidase case, because the active site movement is restricted because of this corresponding covalent bond formation, that means thioether bond formation. The cysteine sulfur, what is coming as S minus, is binding to your histidine residue, giving a new sulfur carbon bond to that of your residue, that imidazole residue, which is coordinating to your copper center. So, that means your ligand part, the immediate ligand part around copper center is getting modified, and due to that modification, basically you have the restriction in the movement. So, this is one side of this ligand coordination, and you are talking about the movement of this particular. So, if you are able to restrict that, your functional property will be changing. That we see then at the in this particular example, that where you have the melanin, where we have the skin color, our skin pigment is produced through melanogenesis. What is that melanogenesis? Melanogenesis is nothing but the oxidation of the amino acid tyrosine followed by polymerization. So, if all these statements are given to you, what you can understand nicely, close your eyes and think it, that we are talking about oxidation, and we are talking about the oxidation of tyrosine molecule, that very useful amino acid molecule, which is a part of the protein chain also, or it can be a separate thing also, So, when you have the oxidation of the amino acid tyrosine followed by polymerization, what can happen? So, this particular oxidation, what sort of oxidation? So, oxidative transformation of the catechol derivatives, we can see if we allow the polymerization reaction. So, you cannot stop that polymerization reaction. So, is the biopolymerization reaction, and melanin is nothing but your biopolymer on your skin or epidermis of the lower part of the skin, not at the upper part, epidermis of your skin, and which is very important. This color is very important for absorbing UVB radiation. That is why the brown, the color brown is also very important. So, that is why the red to dark brown color is also important. People will not suffer the degradation of this particular melanin part and all these things, and the corresponding skin also, they do not suffer from the corresponding skin cancer. So, these things, so what we have understood so far, that you have the corresponding catechol molecule, convert it to quinone molecule, and this quinone molecule you have the para position substitution, you see, and that substitution can further be transferred. You see that 3 arrows, 3 left hand arrows. So, these 3 left hand arrows basically what tells us that you can have different steps of other reactions, such that the amino acid functions, the amine functions can be cyclized to give you something where you can have the indole function, indole with substitution, and indole with the catechol function. So, it is indole 5,6 quinone, and that is very much susceptible for your polymerization reaction. So, you see one part, only a fragmented part, a partial structure of the melanin is a huge structure, but you should be able to identify these indole quinone form one, then in the opposite one. So, one is this, and another one. So, this is basically cross linking, like that of your sheet, from the graphite we get the graphene sheet. So, it is typically a sheet like structure, which is nothing but your melanin pigment, which is forming over our skin basically. So, this is a very important reaction, but this is basically catalyzed by the copper center. So, quickly we see now at the end the catechol oxidase. So, these enzymes basically also catalyze this thing what we have seen. So, the restriction, in another case you do not have the restriction. If you determine the structure, you will find that the active site of the met form of catechol oxidase from the sweet potato I batatas. So, that basically gives us that the copper A and the copper B site you identify, then another histidine nearby. So, you can have the corresponding bond. So, the difference from tyrosinase is that there is a thioether bond. So, that we are discussing so far, that if you have the thioether bond, your catalytic function will be changing. So, you have a thioether bond on one of the histidine ligand of CUA, which is coupled between these two numbers of the cysteine residue and the histidine residues like the tyrosine residues, and the reorientation is now prevented. So, reorientation due to the formation of this thiether bond is stopped and we are not getting this thing, and sometimes the function of this catechol oxidase can be inhibited. If your thing is overactive, sometimes we require the drug molecule or the medicine to inhibit some function. The catechol oxidase, if it is giving some problem and maybe it is genetically also transformed also, so you go for the inhibition. So, phenylthiourea if you take and you take it that particular the native catechol oxidase, crystallize it in presence of that phenylthiourea, and you get a very interesting molecule which was similar to that of your tyrosinase function of the bisphenol or the catechol binding to copper center. So, phenylthiourea can bind in that fashion where sulfur is bridging these two copper centers, and one of the point basically the nitrogen point is coordinating to one of the copper centers in that fashion. So, similar type of coordination. So, there will be a conformational changes at the active site residues, and is the rotation of the aromatic ring of your corresponding amino acid PHE 261, which is your phenylalanine. So, phenylalanine 261 is responsible and restricting. So, that means the secondary interactions can also be important for inhibiting all these things. So, what we have seen therefore, whatever we are talking nothing but the typical enzymatic orthohydroxylation reactions. In organometallic chemistry, in many other parts we also study the orthohydroxylation reactions. They are not only hydroxylation reactions, but also we can go for the other thing, that means the quinone formation, your melanin formation, which are very important in all these studies in the biochemical world. Then not only the your melanin, but the rotten pigment, the pigments for the rotten fruits due to the enzymatic browning, we know the yellow is then browning and then finally the rotten thing. But what we are producing basically due to that oxidation of all these things, we are producing more and more number of your orthoquinone molecules within it. So, you read catechol oxidase nicely in the Wikipedia page and your related other items which is coming from that particular page and the book. Okay. Thank you very much for your attention.